Hey there. How are you?
Hello. I'm good. Tired, but good.
Awesome. Are you like really tired or
A little bit, yeah.
got it? Okay. Um, okay, yeah, so you sent me a question on PCR.
Yes.
O-
Yeah.
Okay. Let me bring it in. I'll open up the whiteboard. All right. So what can you tell me about reverse transcription or yeah, PCR?
So you're using PCR to like try to find out like if you have a specific sequence or like gene in your DNA strand or like in your sample.
Okay.
Um, and if it's a reverse transcription, that means that you may have like RNA and then you're trying to like get like DNA information out of it,
Okay.
perhaps.
All right. So yeah, so you highlighted the reverse transcription, so you were aware that this isn't going to be a regular transcription because a regular transcription would be from DNA to RNA, right?
Normal transcription. Yes.
Right? Like the central dogma.
Yeah.
So if transcription is DNA to RNA, reverse transcription, I'll just write RT, would be turning RNA into DNA, right?
Yeah.
So, so they're asking about this gene fragment that's the target, and they're asking what could be a possible primer for this RT-PCR. So right away, we should know that, um, well, I guess before I say that, I should say that when regular DNA, um, let's say this is like a strand of DNA with its base pairs, well, just its bases. Um, what would happen is you would have an RNA molecule. I'll use dark red for that. An RNA molecule that will be the primer, and then the rest of the DNA molecule can come in and remove that primer. So that's just an ordinary DNA replication. But since we are dealing with reverse transcription PCR, our primer should be DNA. So here I can find like a picture of so yeah, so this is regular DNA. So you're aware of, first step, the helicase unwinds the double helix. Then single strand binding proteins stabilize the unwound parental DNA. And you can see that the leading strand, which is going to be from, so like if I drew something like this, like that, and let's say that this is 5 prime, 3 prime, 3 prime, 5 prime. What I can do, and I'll do it in blue, inside of this would be, well, you are having like a new strand forming in this direction, and it has to start with the 5 prime end. Whereas here, it will replicate in the same direction, but it's starting as a 3 prime. Right?
Yeah. Is that right? Like the different polymerases, they're synthesizing them in the same direction? Or is it just one in one direction and the other in the opposite one?
Oh,
I'm forgetting.
yeah, they're going to be going. So the one that, okay, so the leading strand is synthesized continuously
Mhm.
in the 5-3 direction by DNA polymerase. So that's going from from the new 5 prime end that's being created with it going towards the end where it will become 3 prime. So if we think about that, well, I'll guess I'll get to that in a sec. So now for the lagging strand that's below, this starts off as something that would be 3 prime. But there's something weird that happens there. What do you think?
You can't really synthesize continuously. Like from the so you can't really do it in this direction. So you have to like add only one at a time and then ligate it.
Uh-huh.
I think. Yeah.
So um okay, so the leading strand, um okay, you know what? I think yeah, no, you were right about the whoops. Yeah. About the direction. So, okay, so the leading strand here is just going from 5, 3, whereas the lagging strand is going to go like in this direction. And you can imagine, it's starting off here at the 5 end, so this is going to be the 3 end here.
Yeah.
So, okay, so it says the lagging strand is synthesized discontinuously, where a primase synthesizes a short RNA primer, which is extended by DNA polymerase to form an Okazaki fragment. So why do you think there are lagging strands and leading strands?
Um, because of the directionality of the polymerase
Okay.
when it's synthesizing and like reading.
Yeah. So tell me about the significance of like a 5 prime end and like a 3 prime end.
So like the polymerase uh it's can only like it it can only read upwards, so it can only read from like wait, let me think about that. I know it's um so 3 to 5. So it can only read in the 3 to 5 direction. And it can only like synthesize DNA in the 5-3 direction.
All right, tell me why.
Um, I don't know why. I just know that.
Uh-huh.
Yeah.
So, um, so for a DNA molecule, generally looks like this. This is a base. Um, and then this is a like a phosphate. Um, and there's an OH here for DNA. Now, um, okay, so this is carbon 1, carbon 2, carbon 3, right? Carbon
Mhm.
4, and then I think there's another carbon there. Here, I'll erase this for a sec. Carbon 5, then um then it would be oops. Then it would be yeah, the phosphodiester like that. So okay, so the 5 end is this, that phosphate. Okay. The 3 end refers to this hydroxyl group. Now, when we polymerize nucleotides, how do you think that happens?
So you have to make the phosphodiester bonds.
Okay. What
With
tell me what happens first.
mm,
So there's a certain functional group here that I say every time we see it, I say I want you to think of it as something.
it's a nucleophile,
Yeah, what is?
the phosphate
Mm,
or the alcohol. I don't know.
yeah. Do you remember when
Mm,
like so definitely write this down. So hydroxyl group is the most common nucleophile that you will see on the test. Okay. And the most common electrophile you'll see on the test will be a carbonyl. So if you had an OH group, a carbonyl group, right? This will attack that, go up like that. So definitely remember, this is the nucleophile electrophile, okay?
okay.
Every time you see a hydroxyl group, think nucleophile, okay? Because in fact, if I drew this, oops, here, and I added this, what is this now called?
Like ribose.
Yep, because it's not the deoxy, right? Now, why which one do you think is better? Or
Um,
better? I mean, which one do you think is more stable?
the deoxy one.
Why?
Um, because it's less reactive then.
Yeah, but how? How do you know?
because it has one less OH group.
Wait, sorry. So which one are you talking about? Are you talking about so you said which one is more stable?
The deoxyribose.
Um, well, if it was more stable, why wouldn't we have Oh, sorry, you're saying my bad. You said which one is more stable?
Deoxyribose.
Oh, yes, good. All right, and yeah, why do you think?
Because it's less reactive.
Yeah, why?
Because it doesn't have that hydroxyl group.
Yep, because that hydroxyl group acts as
Um,
acts as what?
a nucleophile.
Yep. So you would see something like that attacking. Um, so that's why RNA is not is very unstable, and it's only produced when you need it to eventually translate into a protein, but it doesn't stick around.
Okay.
So okay, if we go back here, we can we can see that okay, so what happens in polymerization is this hydroxyl. So okay, just like the carbonyl group is a electrophile with a partial positive, partial negative. Phosphate is similar, right? It's a phosphate that's having its electrons being pulled by the oxygens, right? Making it a good electrophile. So when DNA replication happens, the hydroxyl group as um acts as a nucleophile to attack this, and it can then kick something off. But that's how you're going to have your two um, nucleotides attached to one another,
Okay.
right? So, so you can see that if we, so you could see that the 5 end has the phosphate and 3 end has the hydroxyl. And since you need that 3 end hydroxyl to cause the DNA to polymerize, that's why you need it to go in that 5-3 direction. Because in the 5-3 direction, it would be like, think about it like this. Like,
You always have like a free three end with the OH. Okay. I see.
yeah, like it would be like this, like going upwards, because you have
Mhm.
so when these bind, this will be the free 3' end, right?
Mhm.
So that's just why the three um the 3' end is talked about a lot. So over here, from this 5' end that's being produced on the leading strand, it's doing it continuously just fine, right? But for this one over here, you can't have it go in that direction, because if you had it going in that direction, um, it would be uh going from 3' to 5', right?
Yeah.
So it won't go in that direction. It'll go in the direction to the right, and it won't be continuous. So what you have happen is you have RNA primers, which can be added in, a 5' to 3' like direction. So like for instance, if you had like 3' to 5' as your template strand, your primer would be like 5' to 3 prime, and it'll match those bases.
Yeah. And primers, I guess this is a confusing part of this question. The
Mhm.
primer is also made out of, is it like made out of RNA or DNA?
Oh yeah,
Normally.
made out of RNA.
Mhm. Normally out of RNA. Okay.
Yeah,
Okay.
yeah. So you're going to have, yeah. So essentially, you're going to have like in the leading strand. I mean, they didn't really write it, but uh, you can have like one, I guess, primer, but for the lagging strand, that has to be in that 5-3 direction. You have to go in the opposite direction from the replication fork. And um and yeah, that's when the um RNA primers are inserted, and then the polymerase can attach DNA after that, and then those are called Okazaki fragments. And then the last part involves removing that RNA primer and replacing it with DNA.
Yeah. So it's normally RNA primers. And why is it that when you're doing PCR, so normally with PCR, you would also be doing RNA primers, but with reverse transcriptase, you're using DNA primers? Is it like always having to be the opposite? Like if you have an RNA strand, you would need DNA primers, and if you have a DNA strand, you would need RNA primers?
Yeah, yeah. So I mean they would have to say like reverse transcription, because I mean you can do a PCR that uh, that doesn't have to have a DNA primer. But this is just talking about uh specifically reverse transcriptase PCR.
Okay.
So that's the key word here. So that means that, so yeah, so just think of it, I guess, as yeah, think of it as, yeah.
Here it says PCR uses DNA primers. So is it just like a technique thing? Like they just always use DNA primers, always like whether it's RT or normal PCR?
Oh yeah, yeah. So sorry, because I was talking about just replication,
Yeah, yeah.
but
Understand.
for PCR, that's when you like, you artificially um, you know, cause that cause the like overall like unraveling of the helix, and then forming new nucleotides attached to that. So yes, okay. For PCR, um yeah, so for PCR, like the DNA primer that's used, um well, why do you think that is uh what can be used?
Um, why they use DNA primers and not RNA primers in PCR?
Yeah.
Maybe because it is more stable, and so you can keep it better in like a lab, or like before you use it, you can apply it and it won't like it wouldn't react as much, so it's more stable and it won't mess up your experiment. I don't know.
Uh-huh. So if we think about um let's see. Here. I'm trying to find like a good picture. Okay. You know what? Okay, I think I found something. Okay. Yeah. So, uh, this also has something to do with cDNA. Okay. So you were saying that PCR uses DNA primers, so we can eliminate C and D because they contain uracil and are therefore RNA. When a question asks you to figure out a sequence, it's often a good idea to work methodically and write down each step. You usually don't have to transcribe the entire sequence, just a few base pairs should be enough to identify the correct answer. Pay close attention to polarity, 5' ends versus 3' ends. First, we must recognize that the sequence given is the coding sense strand and not the template antisense strand. That means that the mRNA that would be produced has the same sequence as the coding strand, except for the thymines, which become uracils. So it should look like that. Now the mRNA is going to be reverse transcribed into cDNA for use in PCR. And so the cDNA will be like 3', blah, blah, blah. Remember the amplification primer will be complementary to the cDNA sequence. So it's surprise, going to be blah, blah, blah. So, okay. So here we have an example of the um, the RT-PCR to be making cDNA. So we start off with an mRNA up top, and we hybridize it with this primer, DNA primer, that is. And we start to reverse transcribe RNA into cDNA. So we see that happening. And then we have, well, we remove the RNA with something and we add this tail to get our single-stranded cDNA, and the rest of it just proceeds as you would expect. So, yeah, so just think of the PCR as, so okay, think of regular DNA replication as using RNA primers, and the purpose for any primer is to provide a free 3' hydroxyl group, right? So now in terms of PCR, well, if we're doing, well, I guess I should say regular PCR, you're going to be doing DNA primers. But especially for reverse transcriptase PCR, you're going to be, you know, using, since you're turning RNA into DNA or into cDNA, your primer will have to be composed of DNA.
Okay. All right.
Yeah, I have a um so PCR depends on the ability to alternately denature double-stranded DNA molecules and hybridize complementary single strands in a controlled fashion. Starts with heat denaturation of DNA sample into single strands at 95C. Next, two synthetic oligonucleotides complementary to the 3' ends of the DNA segment of interest are added in great excess to the denatured DNA. And the temperature is lowered to 50 to 60 C. The specific oligonucleotides, which are present at a very high concentration, hybridize to their complementary sequences in the DNA sample. Um, so, uh, essentially, let's see. Okay, so I'm just reading from a textbook, by the way. Um, so okay, saying that a variation of the PCR method allows. Here, I will. Okay, so here we're talking about a variation on the PCR method allows PCR amplification of specific DNA, cDNA sequence made from cellular mRNAs. This method, known as reverse transcriptase PCR, begins with the same procedure described previously for isolation of cDNA from a collection of cellular mRNAs, typically an oligo deoxy T. Yeah, primer, which hybridizes to the 3' A tail of the mRNA, is used as the primer for the first strand of cDNA synthesis by reverse transcriptase. A specific cDNA can then be isolated from this complex mixture of cDNAs by PCR amplification using two oligonucleotide primers designed to match sequences at the 5' and 3' ends of the corresponding mRNA. These can include restriction sites and things like that. And it can be performed so that the starting amount of some mRNA can be determined accurately. So you can, yeah, so you can see in this image here, each primer for PCR is complementary to one end of the target sequence. So we have a region to be amplified. So that's in the question stem, the region, right? In the question stem, like AGC, CTT.
Yeah.
So that'll be that region that's to be amplified. Um, and it's 5'3", and this can have a like this DNA primer attached to it, and which would be, so let me write something. Um so this is the like sense strand, right? Like the
Yeah.
coding strand. And then this is the template strand, right?
Yeah.
And if and the template strand is complementary to the sense coding strand. So if you then build DNA that's complementary to the um, template strand, you get your original sense strand again.
Yeah.
So that's what we have here with the primer and then DNA synthesis. And that's just round one, and then they could just do it like multiple times to do it more and more and more. But yeah, I would say that if you can just remember that whenever you have like okay, so the best broad way of looking at this would be that um well, what's the purpose of primers?
Um, to have a free 3' end to be able to nucleophilically attack and like extend your
Perfect, perfect.
sequence.
Yeah, that's perfect. So yeah, so the primer's there because it needs to start it out, which would leave it as a with a 3' end, and then it can further polymerize. Um, and in DNA replication, you have RNA primers, but in PCR, you you continue with the DNA primers.
Okay.
Hmm?
Yeah. I guess if we can go back to the question, I'm still like wrapping my head around the fact that like um it's not like the complementary sequence that we need for this primer, that it's the exact same sequence. It's saying this is a coding strand.
Mhm.
Coding strand. I see. Coding strand would be like the one in the bottom that's lagging. And so we want the template strand, not the coding strand. That's why. And that's why it's identical. Okay, I think that I could, now I see it.
Mm-hmm.
Yeah. Okay. I think I see it. I think it always like confuses me. So let me like mentally review. So like you have the template strands. So I usually envision it at the top. And this template strand is also called the sense strand. Is that correct?
The coding strand will be the sense strand. The anti-sense strand is the template strand.
Why is it the antisense?
Like, why is it like in actuality, or why is it called that?
Yeah, like why is like the sense from 3 to 5 and not from 5 to 3?
Well, usually you'll see it as 5 to 3, but it's basically the coding strand, right? So if you make something, so like our coding strand is that,
Mhm.
and TCG like this would
Yep.
be, well, I guess it would have to go in the 3'. So I guess the one that you chose, let's see. You see, yeah. So what you chose is would be the template strand.
I see.
And the template strand is used to make the next like DNA strand, which would be complementary to the template strand. which would be then the coding strand.
Mhm. Right. So you said the template strand is the antisense strand,
Mm-hmm.
and the coding strand is the sense. Perhaps is the sense you're referring to of like the newly synthesized, like the directionality of the newly synthesized sequence? Is that what they're referring to when they say sense?
Tell me that again about sense.
Is that like the directionality of the newly synthesized sequence that they're referring to, like sense in that in like the same direction? Um, yeah, I'm still wrapping my head around why the template strand is called the antisense strand and not the sense strand.
Oh, so um, I don't know like why it's called that, but you're what you're essentially doing is like let's say we have so the 5' AGC CTT CGG CTG ACT GG whoops C and then 3'. Now this is they're telling us that this is the coding strand, right? And coding is equal to sense strand. Now, what's complementary to this coding strand? So I'll write this in green. So it's going to be U, C, G, G, A, G, C, C, G, A, wait. Yeah. Um, that number C, T, G, okay. C, U, G, A, G, G, or rather C, C, G, 5', right?
Mhm.
So now this is the template, also known as antisense, and it's called a template because that's what you use to to make the next strand. So let's say we have, I'll draw it in purple, like the newly synthesized strand, right? Because they're asking which of the following is a primer that researchers could use for amplification. So we want something that will return to the 5 to 3' direction. So and we can find that by finding out what's complementary to the template strand. So the U turns into then A again, the C turns into a G again, the G turns into a C again, right? C U U C G G C U G C C A G G C, right?
Mhm.
So oh, and then this is the new strand, right? And you see how that new strand is identical to the coding strand?
Yeah.
Oh, I made a mistake. I made it in RNA.
Yeah.
So my bad. All the U's should be T's. Um, but yeah. So your answer choice would be um the template strand. So in addition to C and D being RNA. And see how it says, although RNA primers are used in vivo for DNA replication, PCR uses DNA primers.
Yeah.
So keep that, yeah, keep that in mind.
Okay. Sounds good. Um, yeah. Sounds good. Do you mind if we end early today? I think I want to get home.
Yeah, totally.
Yeah.
I was gonna send here. This is a link of some questions about DNA replication. I was trying to find stuff on um PCRs, but I'm sure you can find some in the UWorld.
Okay.
So try to see if you can. It probably will be under like research techniques or something.
Yeah.
But yeah, look into that. Try those out. Um, and then we'll get a better idea.
Okay.
Yep.
Sounds good.
All
Well,
right. Awesome. So
bye.
I'll see you tomorrow. Okay.
Yeah.
Take care. See ya.